camera on.
All right, it is so good to see all of you here today. Needless to say, Moodle is down, as you have probably discovered. I had no idea that when we did the Zoom meeting setups via email that would be such a great benefit, because some faculty evidently just put their Moodle links on, or their Zoom meeting links on the Moodle site, and now students can't find them. So yay for us for having no foresight whatsoever, but doing the right thing in the end. But I gave my students in my ecology of food class, a CTI class that I teach, I gave an exam this morning, and I revealed, giving the exam without the benefit of Moodle, revealed some problems with Moodle that I hadn't foreseen that I want to just tell you about. So because this is the least preferable way for me to access my email, I didn't even think about this, but there is a subset of students evidently who access their email exclusively through central.jewel.edu, the intranet. And the unfortunate thing about that is that they also couldn't access their email because they didn't have access to their email except through the school's web portal. And as a result, some of them were taking their test on an iPhone, which seems like also a terrible way to go about taking an iPhone. And so if you haven't done this already, and if you're not doing this currently, you should probably just have Outlook loaded onto your iPad and your computer so that you can access the school's mail server without having to go through central.jewel.edu. Because, for example, right now, if you're doing that, you cannot get the email that I sent out a few minutes ago that has today's PowerPoint slides attached to it. So I sent out an email, it has today's PowerPoint slides attached to it. You would already have these, once again, if you downloaded the ANOVA file, the ANOVA PowerPoints that we went through in lab, but I did make some minor changes to the PowerPoints. But if you, once again, were depending upon Moodle to access the ANOVA PowerPoints, once again, you're not going to be able to do that until Moodle comes back up. So just some technological glitches that we could not have foreseen that caused my students some grief earlier this morning. And I learned some things, once again, about kind of the limitations of how people interact with digital information. So anyway, that is that. Dr. Judd, are you on? Can you guys hear me? Yeah, okay. I'm not sure why. Is Dr. Judd on? Where is Dr. Judd? I don't see Dr. Judd. Okay, everybody unmute yourselves for a minute. I just want to get some feedback. How did you guys feel about the exam on Wednesday?
Eh.
What's that, Keaton?
Eh.
Yeah. Well, that could be better.
Yeah.
What were some issues you had?
I just seemed to forget everything I knew.
Okay. Is that a test anxiety thing, or is that just you didn't study well enough? And now I realize I'm kind of putting you on the hot seat because you're the one who answered first.
Probably a little both. And
Okay.
I felt like I was prepared, but sometimes that just happen.
Okay. One of the things that's sometimes useful when you're taking a test is if you get thrown by a question, just move on to the next question. And sometimes what you'll see is as you're going through the test, you'll get to another part of the test, and it's like, oh, this part of the test is related to a different part of the test, and my brain is latching on to this part of the test better than the other part of the test, and then go back to that old question and approach that question again, given that you've seen some of that information on the test in another form. That sometimes will help you answer the first question. Also, it'll keep your anxiety down, because when you get to a question that's stumping you, you don't think to yourself, oh my gosh, I'm stumped on this question, I'm never going to get this. It allows you to free yourself up from that question a little bit, and then just keep going through the test, and maybe something will be triggered as you're going through. The other thing is your subconscious works on things while you're not thinking about them as well. And I was just telling Dr. Judd a story about me making out a test where my subconscious worked on something. I had to go back and change a question on a test because this just wasn't a very fair question for my ecology of food students, given kind of the way we covered the material in class. Dr. Judd, when you are on Zoom, sometimes you have just your face on the screen with your board behind you, and it's big. I cannot figure out how to get Zoom to do that for me. Do you have a hint?
Just my face is big. Oh,
yeah.
In the top right corner, you should have the speaker view versus gallery view option.
Well, I do that sometimes, but it still makes me small. See, you're big on my thing now, but I'm the one speaking.
There's also a speaker.
There's also what? I can't hear you.
Sorry. There's also a speaker view option where it's pinned or unpinned. So if you right click on somebody and click pin video, then it'll stay on them.
Can I do that to myself?
Yes.
Oh, I can do that. Oh, there I am. Okay, awesome. Thank you for that. Okay, but then will they be able to see my screen when I
They will. Yeah, it won't affect them.
Okay. All right. Cool. Okay. Good. Okay. Any other feedback about the exam? You guys got nothing else to say about the exam? All right, then we'll press on as if everything is fine, I guess. All right. Today, we might actually finish early because we've already been through this material in lab, but one of the things that educational research has taught me is that the more times you are exposed to something, the more likely it is that you are going to learn that. So we're going to go through analysis of variance, and then we're also going to talk about the coefficient of variation. So these are both topics that are in the next case study that we're doing. So we spent a lot of time on the Galapagos Islands. Seems like we spent forever on the Galapagos Islands. And what I want you to see now is that we're moving out of that, and we're going to be talking about the Konza Prairie. So the Konza Prairie is this prairie over south of Manhattan, Kansas. It's 8,600 acres of unplowed tallgrass prairie, and they have a long-term ecological research site there that you will be being introduced to by Dr. Judd on Monday, and he's going to have some homework for you, I think, to prepare for Monday. And I'm also going to have some homework for you to prepare, where you're going to run an analysis of variance on some data. I would have uploaded that to Moodle this morning, but Moodle is down now, so I will put that up as soon as I am able to put that up. So can you guys see my PowerPoint at this point?
Yes.
Okay. So In lab, we gave you this example of an experiment. It's a weird experiment. I'm not a botanist, so I can't really wrap my head around this experiment, but basically they watered different pea plants with different kinds of water. Control water that has nothing in it, 2% glucose added to the water, 2% fructose added to the water, 1% glucose and 1% fructose. So you now have two simple sugars, but they still only make up 2% of the water. And then 2% sucrose, which is a double sugar made up of glucose and fructose. But once again, you keep it at 2% concentration. And then they looked and saw how much the plants grew over some period of time. And so the control, 2% glucose, 2% fructose, 2% mixture of the two, and a 2% of a double sugar. When you plot these data out on a number line where the x-axis is the amount of growth, we see that the black is control, the green is glucose added, the red is fructose, the blue is glucose and fructose, and the yellow is sucrose. And if you looked at this, you might assume what? Keaton, since you felt marginally prepared on the last exam, we'll work on getting you prepared by asking you questions.
I don't think we can see what you're. I think it's on a different slide than what you're talking about.
No, why did that pause? All right, let's try this again. Yeah, you guys should stop me when that happens. Can you see it now?
Yes.
Can you see me moving around?
Yeah.
Yes.
Okay,
Yeah.
all right, good. All right, so here's the numbers. So control, glucose, fructose, 1-to-1 and 2%. And this is what the data look like. And when you look at these data, now Keaton, what would you conclude about these data based on what they look like and what you know from how we went through this in lab? Keaton, are you with us?
Sorry, I was muted.
Can you read the x-axis out loud while he's talking?
Yeah, the x-axis. Well, what do you mean?
And what do the numbers mean? The labels
numbers.
by your.
These are the growth increments on the x-axis. So these are the numbers that are on the slide previous. So these are these numbers. And then I've just taken those numbers from control, glucose added, fructose, glucose and fructose, and I've just plotted them above the actual data. So these are what the data look like if the x-axis in this case is the growth increment for these P's. So what would you conclude about this?
Sorry, that the 2% sucrose added has a higher growth increment than any of the other added things other than the control.
Sure, and then it looks like the control is growing faster than everybody else. Good. So we could calculate the averages for all of these things, the sample means for all of these things, and we could calculate the 95% confidence intervals, and I've done that, and this is what they look like. And once again, let's see, who else? Devin Sparks, tell me how do we decide whether or not two means are different from one another?
You said means?
Yeah, how do we tell if two sample means are different from one another? So if we
well, mean is like the average of all of them, right? So
it's like, the average of the sample.
Yes. So I guess
So for example, how do we tell? So Keaton said that it looks like 2% sucrose is different from all of these. So how would we tell that 2% sucrose is different from the 1-to-1 mixture of glucose and fructose? The yellow different from the blue.
Sorry, my iPad's a little messy, so I can't see the sucrose. Okay, so maybe because it only has less than the mean, it was only like 2.
I'm not sure I'm understanding.
Yeah, I don't know how to word it. Honestly,
Uh,
wouldn't.
Dominic,
need
can
to
you
get
help
her.
him out?
Yeah.
Dominic, are you
Always
there?
Little.
You were just unmuted a minute ago. Alexis, help us out.
It's like if they fall within each other's 95% confidence intervals.
Okay, so kind of an intuitive way of thinking about this is you can ask yourself, does this mean fit within this means confidence interval? And does this mean fit within this means confidence interval?
Okay.
And if they do, then chances are they're not different from one another. But if they don't, then the idea is that they came from two different populations, because this is a 95% confidence interval of the sample mean, which means that 95% of the sample means that you would draw from this population would fit in this range. 95% of the sample means that you would draw from this population would fit in this range. And so if this one falls outside of that, then there's a less than 5% chance that those things came from the same underlying statistical population. So that's an intuitive way of doing that, is just ask, is this mean different from that mean? Is this mean different from that mean? Is this mean different from that mean? But unfortunately, what happens when you do that is it's just making a bunch of t-tests. T-tests are this statistical technique that you might have learned about in high school. Generally, when I talk to students about statistics, it's the only test that they learned about in high school. And the problem with doing this is that every statistical test that you do has a type 1 error associated with it. We talked about this some in lab. Who can remind me what a type 1 error is with reference to sample means?
I mean, it's right there on the slide, right? The probability of saying that two means are different when they are not.
Oh, damn, I forwarded too far into it. Yes, it's the probability of having, I was looking at a different screen. It's the probability of saying that two means are different when in actuality they're not. And so it's making a mistake. It's concluding a significant difference when there's not a significant difference. So this is another way to say it with symbols. This is saying that two things are actually different when in actuality they are not. And so you only want to do that at a certain rate. And the type 1 error rate is called alpha. This is the type 1 error rate. And usually we set alpha at 0.05, which is why we calculate 95% confidence intervals. Because the 95% confidence interval tells you where 95% of the sample means will occur. And so there may be some sample means outside of that range, but only 5% of them will be outside of that range. So this is what alpha is. And we get to set what alpha is as experimenters. And this is just kind of an industry standard level of setting alpha at a 5% rate. But what it means is if you do 100 tests, and they're all at 0.49, and you conclude that all of those tests showed significant differences among means, then 5 of those 100 tests were concluded incorrectly, essentially. And so you don't know which ones of those were concluded incorrectly, but at least 5% of them have been, because you chose this particular rate at which you'll make this particular mistake. But the reason that this becomes important, and the reason that we need something like the analysis of variance, is we could go through and we could basically say, is sucrose different from the one-to-one? Is sucrose different from fructose? Is sucrose different from glucose? Is sucrose different from control? Is control different from glucose? Control different from sucrose? Control different from the 1-to-1 mixture, control different from 2% sucrose, et cetera, et cetera. But when we did that, we would actually be doing 10 different t-tests. And the problem with doing 10 different t-tests is that these probabilities of making a type 1 error, they add up. So the probability of a union of events is the sum of their individual probabilities. And so it would be taking this 0.05 and multiplying it times 10. And so our alpha level is now 0.5. So if we were to do all of these things, then our overall alpha level is 50-50. It's a 50% alpha level, which means we have a 50% chance of concluding that 2 of these means are different when they're not. And if we're going to do that, we might as well just not spend the time doing the experiment and just flip a coin on the differences, because that's the level of statistical power that we have for making a conclusion. And we would like to ultimately be better than that. So what we want to do is we want to have a method by which we can compare all of these means simultaneously. So here's some of the stuff that's different from how we covered it in lab. So when we think about an analysis of variance, and I did give you this in lab, ANOVA, the abbreviation ANOVA, is the analysis of variance. And I made the point in lab, why are we using an analysis of variance to make decisions about means? Shouldn't it be an analysis of means? But it's not an analysis of means per se, it's an analysis of different kinds of variation. So there are different kinds of variation. One is the variation between each observation and the overall mean of the entire data set, which we call the grand mean. So that's one type of variation of each individual data point from the grand mean. But there's another type of variation, which is the deviation of each observation from the observation sample mean, which we've already calculated before. That variation is quantified by summing up all of the differences between each of the individual values in a data set minus the sample mean and squaring those deviations. This is the numerator of the variance. This is the sum of the squared deviations of the individual sample data from the sample mean. So it's the sum of the squared deviations of each individual value from the sample mean. That's the second one of these things up here. But the first one of these things is the sum of each of the sample observations from, as they deviate from the grand mean. And the grand mean has 2 bars above it because that's the grand mean squared. So this is the sum of the squared deviations of individual data from the grand mean. But once again, it's a sum of the squared deviations of something from a mean. So in all of these cases, we abbreviate these SS because this is the sum squared deviations from the mean, the sum squared deviations from the grand mean, as opposed to from the sample mean. And then the last one down here at the bottom, the sums squared, in this case, is the difference between each sample mean from the grand mean squared. So it's the sum of the squared deviations of the sample means from the grand mean. Well, this is the sum squares due to the treatment This is sum squares within, and this is sum squares total. And so another way of saying sum squares treatment is the sum squares among means. So you also see this written sum squares among. But it's basically quantifying 3 different forms of variation within this data set. Sum squares among is variation among the treatment means. Sums of squares within is the variation within treatment means, within treatment samples. And sum squares total is the variation of the entire data set. Okay? Is anybody unclear on what I mean by this at this point? Okay, I'm going to go through showing you what this means. I'm going to show you some differences, and I want Casey to tell me what difference it is that I'm talking about. Casey, you ready for this?
Sure.
Okay. Can you see my screen? Can you see my cursor?
Yeah.
It's hovering over the yellow mean.
Yes.
So let's do this. Here's where the grand mean is. So if we calculated all of these data points up and took their average across all treatments, this is the overall mean of all of these data. So Casey, hang with me here. I'm going to show you some deviations, and I want you to tell me what kind of sums of squares I'm talking about. The deviation between this point and the mean, this point and the mean, this point and the mean, that 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 and that, that and that, that and that, that and that, plus that different from that, that different from that, that different from that, that 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 from that. You can stop me at any time.
Would it be the sum of squares within?
That is the
Within
sum
it?
of squares within, exactly, yeah. So I'm looking at the difference between the individual data points and their sample mean. I would square them and I would add them up across this whole data set. Okay, who can I pick on next? Let's see. Hunter, are you there?
Yes, sir.
Okay, I'm going to do the same thing I just did with Casey. I'm going to show you some differences, and I want you to tell me what sums of squares we're dealing with. The difference between this individual point and this mean, this from that, this from that, this from that, that 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 from that, plus this from that, this from that, that 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 from that, all those differences squared. You can stop me at any time, plus the difference between that point and that point and that and that, 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 across all these different samples. I can keep going, but I don't want to.
No.
What kind of
Honestly-
differences are those?
I honestly couldn't tell you.
Okay, call on somebody else in the class who can.
I can't see anyone else in the class.
Oh,
I guess
go to-
Daniel.
Daniel Castillo, you've been
Yep.
tagged. Now, what kind of difference am I talking about?
I'm assuming sum of squares among.
What would sum of squares among be?
That's the sample means minus the total average.
Sample mean minus the grand mean, but I wasn't doing sample mean minus the grand mean. I was doing this point minus the grand mean, this point minus the grand mean, this point minus the grand mean. So that, what would that be?
That'd be total then, right?
That would be total sums of squares. So Angelina, if I then went this mean minus the grand mean, the red mean minus the grand mean, the blue mean minus the grand mean, the yellow mean minus the grand mean, and the black mean minus the grand mean, what would that be?
Would it be among?
That would be sums of squares among, yeah. So these 3 different kinds of variation sum up all of the variation that there is in the data set. Sums of squares total all of these points, as they differ from the grand mean, is made up of two forms of variation. How much each individual variable, each individual observation varies from its sample mean, plus how much these sample means differ from the grand mean. So sums of squares among and sums of squares within should add up to be sums of squares total, because total is the total amount of variation, but the total amount of variation is made up of these two subcomponents of variation. So this is scary. Don't worry about it, but I want to show it to you just so you have reference to it. But what we're basically showing here is this is how you calculate the mean. You add up all of these numbers in treatment 1, you add them up and you divide by the number of observations. To get the sums of squares within, you subtract the mean from each of these observations, square those things, and add them up across the entire data set. This is the sums of squares within each sample, but then to get the whole sums of squares within, wait, where is it? It's right here. You have to add those up across all of the different values within each treatment, but then you also have to add that up across each of the different treatments. In this case, we have 4 treatments. This is the sums of squares among. It's the difference between the sample mean and the grand mean squared. But one of the things that I want you to note here is that there's a little in here. For each of those differences, you have to weight that difference by the sample size. And so I'm going to show you some numbers here in a few minutes where you're going to be like, how did you get this number from these other numbers? Well, it's because I didn't multiply it initially by the sample size. So if you had the mean of sample 1, you had the mean of sample 1 minus the grand mean squared, you would then multiply that times the sample size of sample 1 before you then went and found the mean of sample 2 minus the grand mean. You would multiply that times the total sample size of sample 2 before you went on to then handle sample 3 and sample 4. So it's the sum of the sample size times the squared deviations from the mean. And then the total is just looking at each the grand mean is just averaging up all of these and dividing by the total number. And you would just add sum of squares within and sum of squares among to give you sums of squares total. But you could independently calculate sums of squares total by doing what? Subtracting this value minus the grand mean and squaring it. This value minus the grand mean squaring it. This minus that squared. This minus that squared. This minus that squared. Plus this minus that squared. This minus that squared. This minus that squared. This minus that squared. This minus that squared through the entire data set. Or you can just do sum squares among, sum squares within, and add them together. So this is what an ANOVA table looks like when you see it in kind of symbolic form. You calculate all those sums of squares among, and you divide by the degrees of freedom associated with that, which is 1 minus the total number of treatments that you have. Sums of squares within gets divided by the degrees of freedom associated with the within sample degrees of freedom, which is the total number of observations minus the number of treatments that you have. But in both of these cases, what you're essentially doing, remember that we talked about sums of squares within is the sample mean subtracted from the observation squared. When we calculated S squared, which is the within sample variance that we calculated at the beginning of the semester, we just divided by that by the degrees of freedom of the particular thing that we're that we're trying to quantify here, which is variance. And so this is just essentially what is in this figure as mean squared. It's just the sums of squares within divided by the degrees of freedom. It's the sums of squares among divided by that degree of freedom. So the mean squared in this case is simply just a variance estimate. In the same way that getting the sample variance of any individual sample is just the sum of the squared deviations from the mean divided by the degrees of freedom. So this is the reason that we call it the analysis of variance. We're actually comparing one variance to another variance. So when we calculate an F statistic, we're comparing the variance among the means with the variance within the means. As the variance
Dr.
among the
Klawinski,
means, yeah, go ahead.
I have a question. So what's the freedom thing? What did you talk about the freedom?
Degrees of freedom. It's,
Yeah, what is that again? I just wanted to clarify.
so for a single sample, it's just the sample size of that sample divided by 1.
Oh, okay.
But when we do this, let me erase some of the stuff on the board because now I have a lot going on the board. So in this case, if you look at this figure here, what's the degrees of freedom for within sample variance? The within sample variance is the sum of all the values minus the number of treatments that we have. A is the number of treatments. Well, how do we get to that? Well, if this is the degrees of freedom for a single sample, that's n minus 1. If we do this for sample 1, and we do this again for sample 2, and again for sample 3, and again for sample 4, we have 1, 2, 3, 4. We have 4 different treatments, 4 samples that we're looking at. The degrees of freedom for that is n minus 1. The degrees of this is n minus 1. The degrees of this is n minus 1. And the degree of this is n minus 1. If we add these all together, it's the sum of all of the individual samples, sample sizes, minus 1, 2, 3, 4. The sum of n minus 4. That's the degrees of freedom for all of the within sample variation. So that's where that n sub I minus a comes from. It comes from the denominators of each of the variance estimates for the within sample variation. Does that help?
Yeah, thank you.
Yeah, who is that?
It's me, Ryan. Ryan Wilson.
Okay, all right. Yeah. So sometimes the equations look scary, but it's useful to kind of break them down and say, okay, why is this formula written this way? Well, this is the reason that it is written this way. It's the sum of the sample size of the first sample, the second sample, the third sample, and the fourth sample. That's what the little I subscript is about. And then each of those has 1 taken away from it when you're estimating within sample variation. So sometimes knowing how the formula is derived is good, but it's not required necessarily, because ultimately we do all this stuff via computer, although you're going to work a data set that's due next week that's going to be worked by hand. Any other questions about this? All right, then let's just do this for this particular data set. So, whoa, that wasn't supposed to all pop up all at once like that. I made some changes, and I guess I should have run through it beforehand. So here's the sugar data set. So if we add all of these values up, it equals to 701. If we add all of these up, it equals to 593, 582, 580, and 641. That all added up together equals to 3,097. If we divide 70.1 by 10, which is the sample size, we get the mean, 70.1. If we divide 593 by 10, we get 59.3, 58.2, 58, 64, et cetera, et cetera. If we add all of these up and divide by 5, it'll equal 61.94. But, and this is a big but, the only reason you can add this to that, to that, to that, to that, and divide by 5 is why. Why does that work? And why will it not work under a different data set?
Is it because they have the same sample size?
It's because they have all the same sample size. Who was that?
Michaela.
Oh, Michaela, okay. So they all have the same sample size. If they didn't all have the same sample size, you would have to multiply the sample size times the mean, get that number, take the sample size, multiply times the mean, get that number, multiply the sample size times mean, sample size times mean, sample size times mean, take that total and divide by the total sample size of the whole experiment, and that would give you the grand mean. So don't just add up the means and divide by the number of treatments to get the grand mean, because if sample sizes are different, it will give you a wrong estimate of the grand mean. So those things all added up together. If you add 70.1 times 10, 59.3 times 10, 58.2 times 10, 58 times 10, 64.1 times 10, that would all sum to 3,097. Divide 3,097 by 50, and that gives you the grand mean of 61.94. So sums of squares within doesn't have to be weighted by sample size, because in all cases it's 75 minus 70.1 squared, 67 minus 70.1 squared, 70 minus 70.1 squared, 75 minus 70.1 squared, 65, done again, 71, 67, 67, 76, 68. That, all those deviations squared add up to 142.9, 24.1, 31.6, 18.0, and 28.9. That adds up to 245.5. This is the total, this is the sums of squares within. Sums of squares among is 70.1 minus 61.94 squared is 66.59. 59.3 minus 61.94 squared is 6.97. 58.2 minus 61.94 squared is 13.99. These things all add up to 1,077, except that it's very apparent that these things don't all add up to 1,077, because what do you have to do to each of these squared deviations of the mean?
Multiply by sample size.
You have to multiply them all times the sample size. And when you do that, you will find that it does add up to 1077.32. So if we then take 1077.32 and add 245.5 to it, that should give us 1322.82. Now, how else could we have gotten 1322.82? Audrey.
You could have taken the individual data of each one from the grand mean and squared it.
And squared those. And so you always have an internal check on yourself. If you're concerned that you didn't get the right answer for this and this, they should add up to the same thing that you would get if you took 75 minus 61.94 and squared it, 67 minus 61.94 and squared it, 70 minus that and squared it, 75 for the entire data set. That should also all add up to 1322.82. And trust me, if you do that, it does in this case. So once you have the sums of squares within, some squares among, and some squares total, you can add in your degrees of freedom. The degrees of freedom is 1 minus the number of treatments that you have, which is 5 minus 1 rather, which is 4. The within sample degrees of freedom is 45 because you have 50 observations and you have 5 treatments. So 50 minus 5 is 45. And these should all add up to 1 minus the total sample size, which is 49 because you have 50 observations. So 4 and 45 should add up to 49. Some squares among plus some squares within should add up to the sum squares total. The mean squared value is the sum squares among divided by the degrees of freedom for among sample variation. Mean squared within is the within sample variation divided by the within sample number of degrees of freedom. And F is the mean squared among divided by the mean squared within. And that's a gigantic F statistic. And that means that it has a really small p-value. So P varies from 0 to 1. And so this is caveman speak for how you interpret F statistics and P values. F large, P small. F small, P large. What is P?
Is that the
Cameron.
error rate?
Okay, who said that?
Sorry, Tessa.
Oh yeah, Tessa. You're split across two screens, so I can't see when people speak sometimes. So repeat that again.
Is it the error rate?
It's the observed error rate for this particular data set. So P is the observed type 1 error rate. So in this case, it's like, it's 2 times 10 to the minus 16th, I think. I think there's a misprint. It says minus 6, but I think it's minus 16th, which is the lowest p-value that R will generate. What it's basically saying is that if you say that two or more of these means are different, your chance of making a type 1 error rate is extremely low. Now, you could still make a type 1 error, but it's just really, really extremely low. And you compare P to alpha, and you always want P to be smaller than alpha, because this is the error rate that you don't want to go higher than. You don't want to make a type 1 error more than 5% of the time, so you always want your p-value to be lower than that. So if your criterion for making a decision is a 5% error rate, you want P to be less than 0.05, and so 0.049 would get you there. But what we have in this particular case is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. What we have is an error rate, a type 1 error rate that we observe for this particular data set that is this number, which is way tiny smaller than 0.05. And so all that really tells us is that something in this data set is different than the others. It doesn't tell us which means are different from one another. We would have to use a different set of techniques to do that, what we call post hoc tests after the fact. But this reinforces what we already knew, which is that the control seems to be different from everybody else. These three are different from sucrose, and sucrose is different from the control. So there are 1, 2, 3, 4, 5, 6, 7. There are actually 7 differences of the 10 differences that are possible in this data set. And that's why the p-value is so small, because there are a lot of differences in this data set. Control is different from glucose and fructose and the 1% and the sucrose. Sucrose is different from the control, and it's also different from these 3 as well. So there's actually 1, 2, 3, 4, 5, 6, 7. Oh yeah, I did it right. There's 7 different differences in this data set. And so it's not surprising that the overall analysis of variance became significant in this case. So that's how you do an analysis of variance. Do you have any questions about the mechanics of it? With small data sets, it's really not that big of a deal to run one. They're a little cumbersome, but you can actually do it. And I'm going to actually give you a homework assignment where you will do this on a data set. It's going to have some differences from this data set, and that each treatment will have a different sample size. So you need to remember that you have to weight the sums of squares among by the sample size of the individual square deviations from the grand mean and things like that. But it's actually a smaller data set than this data set is in reality. So you have this information before you to accomplish that. And so given that Dr. Judd is going to give you some preparatory homework for Monday, I think I'm going to actually make this due next Wednesday so that you're not stuck with 2 sets of homework over the weekend, because I do want you to be prepared for class on Monday. And I would rather you prepare for Monday than to practice what we did today, but I will expect it to be submitted to Moodle by by the end of the day on Wednesday, rather. So
if you need access to a computer to do any of this stuff in R, like if you want to practice the T analysis again before you jump into the homework, something like that, make sure you get to us today, just in case we haven't already taken care of that for you. Being able to run through this stuff in R is very useful.
It's useful, but I do not want you to do the analysis that I'm going to give you in R. I want you to do the analysis by hand, because that's how you truly learn how the analysis works. Now, and I think Dr. Judd is about to say this. What was Dr. Judd about to say?
In the future, you will be using R for these kinds of analyses, but so getting comfortable with that now is a good idea. And we have the scripts, and I have a handout that you can come pick up if you want a handout instead, in order to practice using R for these analysis of variances going forward. But to be clear, the homework assignment is by hand, because we want you to do one by hand first.
Yes. Now, you could take that data set and put it into R and check your work and make sure that your hand calculations are giving you the same result. This is what I thought Dr. Judd was going to say,
Right.
and I misguessed. You can put the data into R, analyze the data in R, and it should give you the same result as your hand calculation. So it would be a good way to check that, but you don't have to do that. But Dr. Judd is correct. The sooner you get comfortable with R, the better off you'll ultimately be. Other questions? All right then. So let's talk about Kanza. So this is a map of Kanza, and at Kanza they have these experimental watersheds. So each of these experimental watersheds has a different thing happening to it. So the N stands for native grazers, the 2 stands for how many years there are between burns, and A is a replicate number. So here we have watersheds with bison on them, native grazers that are burned every 2 years. Here's a situation where we have native grazers and it's burned every 20 years. Here's a situation where we have native grazers and it's burned every 4 years. Here's a native grazing watershed where it's burned every year. These watersheds down here are where you have non-native grazers or no grazers grazing in them. And so they've split up this 8,600 plus acre environment into watersheds. And we'll talk about why they're using watersheds as their unit of area, probably on Monday when Dr. Judd takes you through the introduction to Kanza. But what their experimental design is in the Veen et al. Paper. So the reason we're teaching you about ANOVA is because that's what they use to analyze their data in Veen et al. And so in Veen et al, what you have is you have 2 different sets of treatments. You have watersheds that are burned every year and watersheds that are burned on a 4-year cycle. And then on these watersheds, there are bison roaming around. And I actually managed years ago to find a little icon of a bison. But then within these watersheds, they've created exclosures. And I call it an exclosure rather than an enclosure because it's not designed to keep things in, it's designed to keep things out. And it's designed to keep bison out of these areas. So we have burn frequency, which is 1 treatment, 1-year burns versus 4-year burns. And then we have areas that are grazed by bison and then areas that are protected from grazing. So this is one way to think about their experimental design in the Veen et al. Paper. Another way to think about the experimental design in Veen et al. Whoa, what happened? Oh, that's weird. Oh, I know why. That's in the discussion. All right. So there's another way of thinking about this where you make a 2x2 table. And the 2x2 table, and I just lost the screen share, didn't I?
We see the unpresented PowerPoint.
Oh, you're still seeing the unpresented PowerPoint. Okay. Well, that's good to know. Okay, all right. So there's another way of doing this where you can put this into a 2x2 table. And I have this in another PowerPoint, which is a PowerPoint that we use when we discuss the Veen et al. Paper. And I should probably just add it to this PowerPoint before I upload it. But you can think of this as 2 different main effects. There is the fire frequency. And we have 1-year burns and 4-year burns. And we have grazer presence. And this would be bison present or absent. And so we have 1 year with bison, we have 4 years with bison, we have 1 year without bison, and we have 4 years without bison. And so these are all possible combinations of this treatment and that treatment. And so in this case, we have 2 levels of burn frequency. We have 2 levels of bison present. And so the number of degrees of freedom for these 2 treatments would be 1 minus the number of levels in the treatment. And so when you think about degrees of freedom, you can think about this whole thing. So that is that. So that's the basic design of this whole thing. Any questions about Kanza? What we're going to be doing is we're going to be looking at what makes Kanza a prairie rather than something like a forest. And we're going to be looking at what the biotic and abiotic interactions are in this place that determine things like nutrient cycling and succession and things like this. And so it's going to be way more ecology than there is evolution in the next case study. But this is a course about evolution and ecology, so that's the reason for that.
As you work on the homework assignment, so during class, it seems that Moodle has come back online. So I will post, I was going to email that after class, but I'll go ahead and post it on Moodle in just a little bit. You're going to be asked questions about what you see on Google Earth. You'll navigate around on Google Earth. Pay attention to what you're seeing and do more than just guess, right? So it'll say, what are these things that you're looking at? Think about what they are, navigate around. You can learn a little bit about Kanza. You can read online. You have resources, right? Google is your friend. We're going to ask you questions about bison and bison behavior. So once again, you might not know a whole lot about everything bison do besides reproduce. What do you do if you're a bison? What's your behavior? That's going to come in. We're going to talk about lots of stuff with plants. We're going to talk about grazing and also the biology of plants, how they photosynthesize. And we're going to get into the abiotic things that Dr. Quincy was just talking about, like climate and fire and those kinds of things. So that's kind of the landscape of stuff that you're going to explore with your preparatory homework that I'll post on Moodle.
Yeah, and as you're looking at the images on Google Earth, there's also a function in Google Earth where you can get timeline. There's a little clock at the top, and you can click on that, and it will give you historical imagery, so you can go back in time and look at how Kanza has changed. And the other thing I want you to do is pay attention to the vegetation that you see when you're looking at the images that you're seeing from aerial photographs, because that also is important. So the last thing that I need to talk to you about in terms of Veen et al. Is this thing called coefficient of variation. Coefficient of variation is used when you want to test whether populations differ in their variability. But because oftentimes the average value of a particular trait will also be correlated with its variability, then you have to have a way of correcting for the differences in the means. And so when variation is correlated with the mean, you divide the standard deviation by the average value. And so if you think about how much variation there is in the tail length from elephant to elephant, that variation is going to be longer than the longest length of a mouse's tail, which is going to just be smaller in general. But because it's smaller, it's also going to be less variable. And so what we do is we estimate variation by expressing variation as a percentage of the mean. And that then makes it independent of the original unit of measurement. And so what we do is we just take the standard deviation, We divide it by the mean and we multiply it times 100. And then that gives us an estimate of variation that we can compare from one population or one context to another context. There is a modified version of this where you correct that Z value by some measure of sample size. But if you just learn the top one of these, that's sufficient. This idea that you can compare variation from one context to another context by just dividing the standard deviation by the mean and multiplying times 100. That's really as much as you probably need to know about the coefficient of variation. So when you see in Veen et al. The coefficient of variation, this is what they're doing to their data. Okay. Veen et al., you will have a literature summary that you need to write about that comes at the end of this case study, just like there was one at the end of the Galapagos case study. If you had to give advice to your future self about that literature summary, what advice would you give your future self?
Start
Joseph.
working on it now.
Start working on it now. Start reading it now. Yes. If you leave it until the night before to start reading it, it's gonna not be good. We've given you information about the statistics of it so that as you begin reading it, you are equipped to understand what they're doing and maybe why they're doing it. Questions?
Will we get feedback on our Hendry et al. Paper before this paper is due?
Yes, that is definitely our goal.
Thank you.
That would be mean to not give you any feedback on your previous one and then make you turn in a new one.
Never know.
All right, well, have a good weekend and we will see you on Monday.
I actually had a question
Yeah.
regarding the lab. So I looked. We have a draft of the intro and methods due on Tuesday. Is that?
Yeah, we don't have that actually. We need to
Okay.
revise the lab schedule because we've gotten off because of life and redesigning the lab and things like that. So you'll be assigned that and you'll be talked through what we're expecting from that when we assign it. Yeah.
Oh, okay.
Thanks.
All right. Just making sure.
Yeah, thanks for looking at that schedule, but know that that schedule is out of date and Dr. Judd and I have on our list of things to do to update that schedule.
All right. Sounds
Yeah,
good.
the lecture one, but I forgot to update the lab one.
No problem.
Oh,
No problem.
yeah. So you'll know when that's assigned.
All right.
It'll be obvious. All right. Well, Dr. Judd, how was that?
I thought that went pretty well. I didn't mean to steal the thunder from the homework that's by hand, but I am worried that some of my students from lab didn't do what I was hoping they would do in terms of learning ANOVA in lab the other week.
Oh, yeah. Mm-hmm.
So,
well, one of the nice things about covering it in lab and then covering it here is that it gets reinforced again. Like, it's not just a lab thing, it's also a lecture thing now, so.
okay.
All right.
Yeah, that's all. I'm going to go finish getting ready
All
for.
right. See ya.
See ya.